Right now, as you sit here, trillions of ancient organisms inside you are starving, and it's because you've been eating to clean. There exists a strange contradiction at the heart of contemporary nutrition, one that reveals itself not in what we eat, but in what we have systematically removed from our plates in the name of health. We have arrived at a moment where purity has become the guiding principle of dietary choice, where the ideal meal contains the fewest ingredients, the least processing, the most recognizable components. Walk into any marketplace and you will find products advertising their simplicity, their cleanliness, their freedom from anything that sounds remotely chemical or complex. This is marketed as progress, as wisdom, as a return to something more natural and therefore more nourishing. Yet when we examine this impulse through the lens of biology rather than marketing, something troubling emerges. The human digestive system did not evolve in laboratories or sterile kitchens. It developed over millions of years in environments where food was never simple, never pure in the way we now understand purity. Our ancestors consumed plants covered in soil microorganisms, ate fruits in various stages of fermentation, drank water teeming with bacterial life, and stored their foods in conditions that encouraged microbial transformation rather than prevented it. The boundary between food and the microbial world was permeable, sometimes invisible. What we call digestion is not actually a solo performance by human cells and enzymes. It is a collaborative symphony involving thousands of bacterial species, each contributing unique metabolic capabilities that our own genome simply does not possess. These organisms break down complex fibers into short-chain fatty acids that feed our intestinal lining. They synthesize vitamins our bodies cannot manufacture independently. They train our immune systems to distinguish between threat and benign presence. They communicate with our nervous systems through chemical signals that influence everything, from mood to inflammation to the integrity of the blood-brain barrier. When we simplify our diets in pursuit of cleanliness, we inadvertently create a biological dessert. A plate containing only chicken breast, white rice, and steamed broccoli may seem virtuous in its plainness, but from the perspective of the microbiome, it represents poverty. These organisms require diversity, complexity, and yes, even a degree of what we might call They thrive on resistant starches, on polyphenols from deeply pigmented foods, on the modified proteins and bioactive compounds that emerge when foods undergo transformation through time and microbial action. The irony deepens when we consider that many of the foods we now view with suspicion, fermented vegetables, aged cheeses, cultured dairy, naturally leavened breads, were once dietary staples, precisely because they solved problems of preservation and nutrition. That's simple. Fresh foods could not. Our great-grandparents did not eat this way out of ignorance. They ate this way because their bodies, like ours, were designed for symbiosis with the microbial world, not separation from it. The ecosystem living inside your intestinal tract operates according to a logic that most of us never consider when we select our meals. These microorganisms are not passive passengers. They are metabolically active entities with their own nutritional requirements, their own preferences, their own capacity for abundance or decline. When we feed ourselves, we are simultaneously feeding them, and the quality of that nourishment determines far more about our health than we have historically understood. Consider what happens when the diet becomes monotonous and stripped of complexity. The bacterial populations that specialize in breaking down diverse plant fibers begin to dwindle. Species that thrive on polyphenols and resistant starches find themselves without substrate. The microbiome, which functions optimally as a rich tapestry of metabolic diversity, begins to simplify into something more barren. This is not a metaphorical starvation, it is a literal one, measured in shifting populations and diminishing metabolic output. When these organisms lack their preferred food sources, they do not simply go dormant. They begin consuming the next available resource, which happens to be the mucus layer that protects your intestinal lining. This protective barrier, normally several cell layers thick, becomes compromised. The tight junctions between intestinal cells, which regulate what passes from gut to bloodstream, begin to loosen. Partially digested food particles, bacterial fragments, and inflammatory compounds that should remain contained within the digestive tract start appearing in places they do not belong. Your immune system, encountering these molecules in the bloodstream, responds as it must. It interprets their presence as invasion, as threat, as reason for alarm. Inflammation becomes chronic rather than acute. a constant low-grade activation that spreads throughout the body. This is not the dramatic inflammation of an infected wound or a twisted ankle. It is subtler, more insidious, manifesting as fatigue, brain fog, joint pain, skin conditions, mood disturbances. The medical system often treats these symptoms individually, never recognizing their common origin in a starving microbiome. The connection extends even further into territory that seems impossibly distant from dietary choice. Those same bacterial populations produce neurotransmitter precursors that travel along the vagus nerve to the brain. They manufacture compounds that modulate the production of serotonin, dopamine, and gamma-aminobutyric acid. When microbial diversity collapses, so does this chemical communication system. Anxiety, depression, and cognitive decline can have roots not in psychology alone, but in the impoverished soil of an undernourished gut. What makes this particularly tragic is that the solution has existed for thousands of years in food traditions we have abandoned. The bacteria starving inside us are the same species that thrive in fermented vegetables, in yogurt rich with living cultures, in properly prepared whole grains that have been soaked or sprouted. They need the prebiotic fibers found in foods like garlic, onions, Jerusalem artichokes, and underripe bananas. They flourish on the polyphenols present in colorful vegetables, herbs, spices, and fermented teas. We have created a famine in the most intimate ecosystem we possess, and we have done so while believing we were pursuing health through dietary. For most of human history, fermentation was not a culinary choice or a wellness trend. It was a survival technology, a way of preserving harvests through seasons of scarcity, of making food safer and more digestible, of transforming the borderline toxic into the nourishing. Our ancestors did not understand the microbiology, but they observed the effects. Milk left in certain conditions became yogurt that could be stored longer and caused fewer digestive complaints. Cabbage submerged in brine became sauerkraut that sailors could eat months into ocean voyages without developing scurvy. Grains soaked and allowed to ferment became breads and porridges that did not cause the stomach distress of their unfermented counterparts. What we now understand through microscopy and biochemical analysis is that these traditional processes were performing transformations of remarkable sophistication. When bacteria and yeasts colonize foods, they do not simply preserve them, they fundamentally alter their nutritional architecture. Complex proteins get broken down into more bioavailable amino acids and peptides. Antinutrients like phytic acid, which binds minerals and prevents their absorption, get degraded, liberating zinc, iron, magnesium, and calcium that would otherwise pass through the body unused. Vitamins get synthesized that were not present in the original ingredients, particularly B vitamins, including the precious B12 that plant-based diets typically lack. The microbial metabolism at work during fermentation produces an entire library of bioactive compounds that fresh foods simply cannot offer. Short-chain fatty acids like butyrate emerge, which serve as the preferred fuel for the cells lining your colon and possess potent anti-inflammatory properties. Conjugated linoleic acids form, which research suggests may influence everything from immune function to body composition. Peptides with opioid-like activity appear in fermented dairy, potentially explaining why these foods have been associated with feelings of calm and satisfaction across cultures. Perhaps most importantly, fermented foods arrive in your digestive system already populated with living microorganisms, many of which can take up temporary residence in your gut or at minimum interact beneficially with your resident populations as they pass through. This is fundamentally different from taking a probiotic capsule containing a few isolated strains. Traditional ferments like kimchi or naturally fermented pickles can contain hundreds of bacterial and yeast species in dynamic relationship with one another, creating a microbial complexity that no supplement can replicate. There is also something profound happening at the level of chemical diversity. A fresh cabbage contains perhaps hundreds of distinct phytochemicals. That same cabbage, fermented for weeks, contains thousands. The bacterial enzymes unlock compounds trapped within cell walls, transform molecules into more bioactive forms, create entirely new structures through metabolic processes our own cells cannot perform. When you eat this fermented cabbage, you are consuming not just vegetables and bacteria, but the entire chemical library they have created together. This explains why populations that maintained traditional fermentation practices, even when their diets were otherwise limited by geography or season, often displayed remarkable health resilience. The Korean consumption of kimchi, the Scandinavian reliance on fermented fish and dairy, the Eastern European tradition of fermented vegetables and kefir, the Japanese incorporation of miso and natto, these were not. The human body is not designed for chemical monotony. Every cell, every enzyme system, every detoxification pathway evolved in response to an environment of overwhelming molecular diversity. Our ancestors encountered thousands of different plant compounds in a single season, each one presenting a mild challenge to metabolic machinery, each one requiring the body to activate specific response pathways, to upregulate certain genes, to produce particular enzymes. This constant low-level stress was not harmful. It was conditioning, the biological equivalent of varied physical exercise that builds strength through adaptive response. When we eat the same 15 foods repeatedly, when we choose only the mildest tasting vegetables, when we avoid anything bitter or pungent or fermented, we deprive ourselves of this metabolic gymnasium, the liver's detoxification systems, designed to process hundreds of different plant alkaloids and phenolic compounds, begin to atrophy from disuse. The cellular stress response pathways that protect against oxidative damage and inflammation lose their sensitivity. We become metabolically fragile, unable to handle even minor deviations from our narrow dietary routine. Consider the principle of hormesis, where small doses of biological stressors actually strengthen organisms rather than weaken them. The bitter compounds in cruciferous vegetables activate pathways that enhance cellular cleanup and DNA repair. The polyphenols in fermented foods trigger antioxidant production, far exceeding what those compounds could provide directly. The organic acids produced during fermentation challenge cellular pH regulation in ways that ultimately improve resilience. Even the temporary inflammatory response to certain fermented compounds can train the immune system toward more appropriate, measured reactions. What we call a clean diet often produces metabolically inflexible humans. People whose bodies panic at the introduction of anything unfamiliar, whose digestive systems struggle with foods outside their narrow comfort zone, whose immune systems overreact to harmless substances because they have lost the capacity for nuanced response. True metabolic health requires exposure to chemical complexity, to the messy diversity of compounds that fermented and varied whole foods provide. We stand at a peculiar junction in human history where we possess both unprecedented knowledge of microbial ecology and unprecedented disconnection from it in practice. The scientific literature reveals with increasing clarity that human health cannot be separated from microbial health, that our bodies function not as autonomous units, but as ecosystems, requiring constant partnership with bacterial life. Yet our food systems have moved systematically toward sterilization, simplification, and separation from the very organisms we need most. Rebuilding this symbiosis does not require dramatic reinvention. It requires remembering what traditional cultures never forgot. Begin with small introductions of living foods into daily eating patterns. A spoonful of traditionally fermented sauerkraut contains more beneficial bacteria than most probiotic supplements, and it delivers them in a matrix of fiber and bioactive compounds that support their survival and function. Unsweetened yogurt or kefir, made from whole milk and containing actual living cultures, introduces lactic acid bacteria while providing the fats and proteins they use as scaffolding in the gut. The process should be gradual because a depleted microbiome cannot immediately handle abundance. Start with small portions. Allow the ecosystem to rebuild its capacity. Then expand variety and quantity over weeks and months. Introduce different fermented vegetables. Experiment with miso in soups. Try kombucha or water kefir. Explore naturally leavened sourdough bread, where bacterial action has already broken down difficult proteins and starches. Equally important is feeding the microbes already present through prebiotic-rich foods. Jerusalem artichokes, garlic, onions, leeks, asparagus, and underripe bananas all contain fibers that human enzymes cannot digest, but bacterial enzymes dismantle readily. These become food specifically for beneficial populations, allowing them to outcompete less helpful species through simple abundance of resources. This is not about perfection or purity. Ironically, it is about embracing biological complexity, about recognizing that health emerges not from isolation and sterilization, but from rich, diverse, dynamic relationship with the microbial world that shaped us